This video is part 1 of chapter 2, Neuroscience, for PSY 110S, the Psychological Sciences. In this chapter, we'll be looking at the field broadly of neuroscience and defining what it is. We will also be looking at the structure and function of neurons, including neuronal transmission and communication within the brain and nervous system. We'll look at the structure and function of both the nervous and endocrine systems, and then take a deeper look at the structure and function of the brain. So first, what is this field called neuroscience? As your authors define it, neuroscience is the scientific study of the brain and nervous system. Historically, if you go back across the history of psychology, we didn't always have a field called neuroscience. We had a lot of separate research areas coming from multiple disciplines such as biology, chemistry, medicine, specifically neurology, as well as psychologists who were studying what used to be called physiological psychology. So today what we have is a multidisciplinary integrated science called neuroscience. And you'll have professionals who work in this space who have psychology degrees, some have medical degrees and specialize in neurology, some are biologists, some are chemists, some are specifically neurochemists. So there's a lot of different contributing areas of science, and I think that's what makes the field of neuroscience so interesting and exciting. As your authors note, the brain really is the control center for pretty much everything that we do. It's responsible for everything from how we perceive our environment and how we make sense of it to how we think and solve problems and remember things. It also is basically the thing that houses our personalities and tells us who we are and what we like and what we don't like, and so on. That's basically what makes us human. The brain has been estimated to have a billion cells, millions and millions of neurons that make us what we are. We start life with, the base development of the brain and the nervous system, and over the course of childhood and adolescence, the brain continues to grow and change and develop and become what it will stabilize to be in our adulthood. But it doesn't stop changing at that point. The brain and the nervous system continue to change across the lifespan. As we experience things, our experiences have an impact on the way the brain functions, and the way the brain functions has an impact on what we experience. Each neuron within the brain and the wider nervous system is poised to take in information from thousands of other cells. And it really is an integrated network. I would argue that no matter how smart artificial intelligence gets, it really is not going to be able to match the complexity of the way in which the human brain and the brains of many other complex creatures, like the brains of, say, some of the aquatic mammals like whales and dolphins, and our relatives, the other primates, have complex brains and nervous systems. To understand how the brain and nervous system work, you have to start with the cellular level. So we'll start with the neuron. If we want to understand how we think, how we feel, how we act, we need to look at that cellular level. Everything that we think and feel and learn and remember all comes from basic neuronal activity. So the cells that are responsible for all of that information transmission is the neuron. Neurons come in a lot of different shapes and sizes. Some of them are microscopically tiny. Others are easily seen with the naked eye. It depends on where they are in the nervous system, even where they are in the brain, and what their specific purpose is. Different kinds of neurons are engineered to do different things for us. There's also within, the whole nervous system, there are other cells that perform support roles. Glial cells have a specific job to support neurons. The difference in numbers between among neurons is about 1 to 1. While there have been some theories that have argued that there are far more glial cells than there are neurons, there isn't really research evidence to support that. What glial cells do is basically create the right environment. They take care of disposing of cellular waste because neurons are a biological thing. They produce waste in the work that they do. So the glial cells have an important job of cleaning that up and helping to maintain what your authors call a stable chemical environment. They also provide a certain degree of insulation and protection for those fragile neurons in the nervous system. Now, this graphic that comes from your textbook, I just want to walk through it with you and so that you have a visual kind of memory for what neurons look like structurally. Recognize that this is an artist's rendering. Neurons, as I said before, come in all shapes and sizes. None of them will really look exactly like this. There are some neurons that have this appearance, but they're never going to look exactly the same. This particular neuron that's pictured here is a myelinated, has a myelinated axon, and I'll talk about that more in a minute. Start with the dendrites. The dendrites, which are these sort of tree-like branch structures emanating from what's called the cell body. The cell body has a nucleus that holds the genetic information for that particular cell. Essentially, and this is, an oversimplified explanation, but it has the instructions for that particular neuron in terms of what its job is. The dendrites bring in information, so they receive stimulation on these little ends from other neurons, and then send that information electrically through the cell body. And then the electrical signal travels across the axon. In a myelinated neuron, the electrical signal will jump from space to space across these little myelinated areas of the axon. The electrical impulse, it's going to travel down through that cell or that axon and then continue on to the axon terminals. At that point, what happens is shifts to a chemical transmission of message across the space from an axon to a dendrite of another cell. So to go over this a little bit more, the dendrites are fibers that project out from the cell body, and they take in information from other neurons. The cell body contains the nucleus and all the other kind of biological pieces, parts, that's such a technical term, that keep the cell alive and keep it functioning the way it's supposed to function. Axons transmit electrical messages down the cell. Some axons are really, really long. They have a lot of messaging that has to travel quite a distance. So, for example, if you could have an axon that travels from the spinal cord out to the muscular system, and it can be quite long. So it has to transmit that message very efficiently in order for the message to be sent and then triggering the next message down the line. Axon terminals are at the ends of the axons, and that's when you transition from an electrical stimulus across the cell body and down the axon to a chemical message, where neurotransmitters, once that electrical message reaches the axon terminal, it triggers either the release of the chemical messengers, the neurotransmitters, or the restriction of them, depending on the type of message that's been sent. Now, this video is just another way of helping you to visualize and understand how neurons communicate with one another. This is the first video that I've used in one of my presentations for your class. Please understand that the way these recordings work, embedded videos in a PowerPoint, the audio doesn't always get picked up very well. So on the YouTube playlist for your class, every one of the videos that I use in my presentations is also going to be in the playlist. So if you have trouble hearing these videos, please watch them again on the playlist.
Neurons communicate with each other, relaying messages throughout your body and powering all of your thoughts and actions. Neurons talk to each other using both electrical and chemical signals. Messages start as electrical signals traveling rapidly down a neuron. These signals are called action potentials. When they reach the gap between two neurons, the messages need some help to get across. The information is transformed from an action potential into a chemical message, which crosses the gap called a synapse. The release of those chemical messengers can trigger an action potential in the neuron on the other side of the synapse, conveying the message onward. Or it can quiet the message. This happens over and over and over. And with repeated activity, the synapse gets stronger, so the next message is more likely to get through. That way, neurons learn to pass on important messages and ignore the rest. This is how our brains learn and adapt to an ever-changing world. Neurons.
So that gives you kind of the basics, and I tend to be very visual. It helps me to remember things when I have seen them as they function. So let's dig a little deeper into neuronal communication. Within, as I've been describing it, within the neuron, communication is essentially electrical. If you take a neuroscience class with Dr. Knapp, for example, you'll learn about this in much more detail, thinking about exactly what is the chemical nature of that transmission. We don't need to do that in an introductory level class, but suffice it to say that the transmission from the cell body down the axon is electrical in nature. Communication between neurons, so across that synaptic space, is chemical. And what's being released into the synapse from one cell and then communicating with another cell is neurotransmitters, which are little chemical messengers. In this smaller offset image that I've included here in the presentation, you can see what's called a synaptic vesicle, and the little red dots represent neurotransmitters. Real neurotransmitters won't look like a red dot, but for the purposes of education, that's what they are. So when the chemical or when the electrical signal reaches the end of the axon, at the axon terminal, it can signal these synaptic vesicles to migrate toward the very, very edge and then to open up into the synaptic space between cells and deposit their neurotransmitters into that space. And that space is referred to as the synaptic cleft. Then you have receptor cells that are designed to receive messages from specifically shaped neurotransmitters. So let's say the neurotransmitter is serotonin. It has its own specific shape, and it will fit, like a lock into a key, a specific receptor designed to receive serotonin. It won't respond to other kinds of neurotransmitters, for example. So that's kind of the nature of the communication between them. Now the electrical impulse...
A nerve impulse is an
Sorry, I didn't mean to click that right away. So the electrical impulse, just to give you more information about that, and I have another video clip here to describe the process of that electrical impulse. So I'll start that again.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.
So that's basically, a short tutorial on how the electrical impulse functions as messages go from the cell body down the axon to the axon terminals. So the information within dendrites can take two different forms. It can either be excitatory, which basically means it tells the neuron to produce an electrical impulse. It says go, go, go. Or it can be inhibitory. It can tell the neuron not to generate the electrical impulse. So basically it's saying stop, stop, stop. So sometimes in your brain you need action to occur. So you need a message to actively be sent, telling the brain to do something. Sometimes though you need to turn something off. So for example, let's say you're driving and you have to slam on your brakes really fast. So an excitatory message would be sent to do the actions that are required to slam on the brakes. At the same time, you'll have lots of arousal and so on. Eventually, the brain is going to have to say, okay, you can stop doing those things. So it's going to send inhibitory messages to turn off certain responses. The electrical impulse itself is basically all or nothing. It either happens or it doesn't happen. There's not a weak impulse or a strong impulse. It's either an impulse or no impulse. Stimuli that vary in intensities, they're encoded based on the quantity, the number of neurons that generate impulses. And sometimes what it takes for a given cell to fire, as it's called, so to actually produce an electrical impulse, it may require messages from many other cells to accumulate and reach that threshold for sending a message. It really depends on the type of cell, exactly what kind of intensity is going to be required for that stimulus to be sent. Now the myelin sheath, I mentioned that earlier when we were looking at those drawings of various kinds of neurons. Some neurons have myelinated axons, and the myelin is basically this fatty substance in those drawings that I showed you from both your textbook and from an external source. It kind of looks like a chain of sausages, and basically the myelin is an insulator, And the purpose that it serves is increasing the efficiency of message transmission. It makes the transmission of the electrical message down the axon faster. So basically the electrical message jumps from non-myelinated space to non-myelinated space along the axon. So it's kind of like the way in which, when you're running, you step, and that makes it faster than if you had to creep along, putting your feet directly in front of each other and cover every tiny bit of space from point A to point B. Instead, you're jumping, and you're faster that way. Myelin, for want of a more clear explanation, does basically the same thing.
Multiple sclerosis, or MS, is a progressive disease of the central nervous system in adults, affecting around 2.5 million people worldwide. MS affects the ability of nerve cells in the brain and spinal cord to communicate with each other effectively. Nerve cells communicate by sending electrical signals down long fibers called axons, which are contained within an insulating substance called myelin. Evidence suggests that MS results from an autoimmune reaction in which a malfunctioning immune system produces T cells that react with and damage the body's own cells. In MS, an unknown trigger activates helper T cells. This enables the T cells to adhere to and cross over the blood-brain barrier, which normally prohibits the flow of substances into the brain. Antigen-specific receptors on the surface of T cells recognize central nervous system myelin as an antigen. The T cells attack the myelin that surrounds the axons. This causes the central nervous system to lose some of its ability to send signals throughout the body, causing the debilitating symptoms of MS. Almost any neurological symptom can appear with the disease, and often progresses to physical and cognitive disability. Permanent neurological problems often occur, especially as the disease advances. After injury, the central nervous system can repair itself. However, after repeated inflammatory attacks, this repair mechanism can become less successful. Current treatments primarily target the inflammatory elements of the disease and are somewhat effective. Innovative treatments are required that can directly target the central nervous system, not only reducing inflammation, but also inducing repair of the damaged tissue.
So I inserted that video here for you about MS because to me it helps me to understand the purpose of myelin by showing you what can happen when it is no longer functioning the way it's supposed to. In MS, this autoimmune disorder and the inflammation associated with it allows T cells that are part of the the immune system to attack the myelin as if it was something dangerous. When that myelin is destroyed, it interferes with and sometimes causes messaging to completely cease from cell to cell. So for example, if you lose myelination that is related to your ability to walk, then you will have loss of function in terms of locomotion. If it's myelination in the systems that are related to vision, you can lose vision, and so on. So the myelin sheath allows, as I said before, the electrical message to be quicker as it's traveling across that axon. When it is destroyed, there are massive losses of function. It also is what is responsible for giving the appearance difference in different areas of the brain between what's called white matter and gray matter. The myelinated areas of the brain are going to be whiter, whereas the non-myelinated are going to be gray. So as in multiple sclerosis, impulses get slower much less efficient, and they sometimes stop when the myelinated areas of the brain are damaged. The chemical communication between neurons, I've mentioned this before, is done through neurotransmitters. Neurotransmitters are chemicals that are housed in the axon terminals, and they are specialized in transmitting specific kinds of information from neuron to neuron. They're each responsible for different kinds of things. In some cases, they overlap in terms of what they do, and sometimes they interact, meaning that they combine efforts to achieve certain goals, such as emotional regulation, for example. The synaptic gap is a space, a gap across which the neurotransmitters are sent, or they're, the way my physio instructor put it, they explode and they dump the neurotransmitters into the gap, the synaptic vesicles. Like a balloon, they just sort of open and go pop and spread that stuff into the synapse so that they can meet up with waiting receptors. This graphic is contained in your textbook, so I put it here as a reminder to study it so that you can understand the various pieces, parts. So synaptic communication starts with an electrical impulse that's traveling down the axon to the terminals. And then the second part is when the electrical impulse reaches that terminal, it's going to trigger the vesicles to migrate to the end of the axon terminal and open, depositing the neurotransmitters into the gap. And that's going to link up with the receptor sites that are on the next cell to continue the message going forward. Now, brain scans, as your authors describe them, they can give us some information about how the brain is functioning, and especially now that we have brain scans like functional MRIs, we can kind of watch the way the brain is consuming energy as it works in real time. Brain scans of certain kinds work because neurons, they're alive, they're living things that need energy. So they require oxygen and other kinds of nutrients like glucose or sugar in order to do what they do. And brain scans can pick that up and show us what parts of the brain are active during a given task or during a given experience. There are several kinds of brain scans. There are PET scans, or positron emission tomography scans, where the person is given a dose through an IV of a radioactive glucose, and the scanning device can pick that up and watch kind of over time what areas of the brain are using that sugar at that point in time. Functional magnetic resonance imaging, or fMRI scans, detect active areas of the brain which are responding based on how much oxygen they're using. And fMRIs have really been a game changer for neuroscience because we can put people in an fMRI machine. They're awake, they're conscious, and we can give them tasks to do, and then we can watch how their brains respond. There have actually been fMRI studies done with dogs as well. They have to specially train the dogs to lay still and be quiet as they're doing tasks. Usually it's visual to tasks that they can be exposed to, and then they can respond. So that's been a real game changer for researchers when they want to look at a living brain as it's functioning and as it's doing its work. And that concludes part 1 of my coverage of the neuroscience chapter, chapter 2 for PSY 110, the psychological sciences.